Dr. Crum. So today we'll be answering the 2 questions on our exam 3 that you posted. So I'll be doing the first one, which is 6A, and it's to illustrate what occurs in mitosis from a diploid cell. So you have interphase, which is where a cell spends majority of its time. Most cellular contents are synthesized continuously during interphase. Amount of nuclear DNA doubles during S phase. There's 2 time gaps. There's G1 and G2. G1 separates S phase from M phase, and G2 separates the end of S phase from the onset of the next M phase. So the first step in mitosis is prophase. So majority of my drawings came from figure 24-1. So here's prophase. You have the chromosomes right here, which consist of 2 sister chromatids. You have the microtubules are starting to form between the centrosomes. The nucleolus is starting to disappear right here, and then you have centromeres that form like in the middle of the sister chromatids. So a cell is in prophase when the individual chromosomes have condensed to the point of being visible as discrete objects as seen in a microscope. Sorry, I'm trying to line up the picture so you can see it. Each chromosome is made up of 2 sister chromatids that are tightly attached to each other. Centrosomes function as the microtubules originating center, where microtubules are assembled and anchored. As centrosomes move apart, they act as a nucleation site for microtubule assembly, and the region between the 2 begins to fill with microtubules destined to form the mitotic spindle, which is a structure that distributes the chromosomes to daughter cells. So as you can see here, this is the mitotic spindle that's starting to be formed right here. So the next phase is prometaphase, which is right here. So you have your astral fibers right here around the centrosomes. You have your kinetochore, which is right here. You have the spindle poles here and here. And then these are just the fragments of nuclear envelopes that are starting to disappear. And then you have the mitotic spindle, or these microtubule fibers right here. So the prometaphase is marked by fragmentation of the nuclear envelope, as I stated right here. Centrosomes movement to the opposite poles complete, and the spindle mitotic microtubules make contact with chromosomes. Spindle microtubules are destined to attach to the centromere on the chromatids via the kinetochore, which is that right there. Forces exerted by the kinetochore microtubules then gradually move the chromatids towards the center of the cell. So this movement towards the center of a cell takes place majority in metaphase. As you can see, the sister chromatids are moved along the metaphase plate. Moved along the metaphase plate, so they're kind of lined up evenly. In metaphase, when the fully condensed chromosomes all become aligned at the metaphase plate, the 2 sister chromatids of each chromosome are already being actively tugged, towards opposite poles. Sorry, I just ate lunch. So what's happening is that each end is going to be pulled a different way. Like this end might be pulled here, and this end might be pulled this way. And so you're going to see how it lines up in the next picture. So the next step is anaphase, which is when the sister chromatids separate. And as I stated, they are facing towards different opposite poles. So anaphase is actually the shortest phase, and it lasts a few minutes. At the beginning, 2 sister chromatids separate and begin moving towards our opposite spindle poles at a rate of 1 micrometer per minute. There are 2 types of movement. There's anaphase A, which chromosomes are pulled centromere first towards spindle poles as kinetochore microtubules get shorter and shorter. And then there's anaphase B, which the poles themselves move away from each other as polar microtubules lengthen. So the last phase is telophase. And as you can see, the sister chromatids have reached the poles, and it's starting to form two daughter cells. So at the beginning, the daughter chromosomes have arrived at the poles, as I stated. Next, chromosomes uncoil into the extended fibers typical of interphase chromatin nucleoli develop, and the NOC on the DNA spindle disassembles, and nuclear envelopes form around the two groups of daughter chromosomes. So basically, as you can see, the nuclear envelope fragments are starting to form along. To form the nuclear envelope, you have the nucleolus starting to appear again. These are the chromatin decoiling into what is seen in interphase, and then this right here is the cleavage furrow. So in cytokinesis, this is going to separate completely into two daughter cells, and instead of drawing another picture, I figured it'd just be easier, like right here, you would have two daughter cells. So that is the end of mitosis. So I'll go on ahead and answer the next question, which is about meiosis, and then there's a couple of questions throughout the question, but I'm not going to repeat them. So meiosis 1 is the reduction division of meiosis because it reduces chromosome, sorry, I can't even read my own handwriting. It's just known as the reduction division. It's the same basic steps as mitosis, but there is no prometaphase. So some differences between mitosis and meiosis 1, or meiosis in general, is meiosis has no prometaphase. Prophase 1 is much longer and complicated than prophase in mitosis, and normal interphase does not intervene between the two meiotic divisions. If it does happen to occur between meiosis 1 and meiosis 2, it's very short and it does not include DNA replication. So the drawing in the book for meiosis was not very detailed. So it's, like I said, it's not going to be a very good drawing, but I tried to do my best at explaining what occurs within each one, so it kind of makes up for the lack of detail in the drawing. One second. Okay, so the first you have prophase 1, and like I said, the drawing was not very detailed. So prophase 1 is very long and complex phase. There's 5 different phases within it. So the Lipotine phase begins with condensation of chromatin fibers into long thread-like structures. You then have the zygotine phase, which homologous chromosomes become closely paired by the process of synapsis to form bivalents, which you can see here. These are the bivalents. The Pachytene phase is where crossing over occurs and is marked by a dramatic compacting process that reduces each chromosome to less than a quarter of its previous length. The next one is the diplotene phase, which is homologous chromosomes of each bivalent begin to separate from each other, particularly near centromere. However, the two chromosomes of each homologous pair remain attached via the chiasmata, which is produced via crossing over. So the chiasmata is basically the sharing of the DNA of that occurs during crossing over, and it's kind of like a bridge between the two. So that's how they kind of stay together. Diakinesis is the final stage of prophase 1. The chromosome recondenses to their maximally compacted state, nucleoli disappear, spindle forms, and nuclear envelope breaks down, making end of prophase 1. So later in the book it talks about synaptonemal complex, and what it states is that it basically is what holds the homologous chromosomes in tight apposition during synapsis formation, which is when the bivalents form. So another question that you asked was what causes significant genetic variability within meiosis, and so crossing over is what causes that, because it's the physical exchange of genetic information between corresponding regions of homologous chromosomes that account for genetic recombination. So you're mixing the two DNA together of the sister chromatids, so you're having genetic variability between different. I'm sorry, I can't think of the word, but you know what I mean. So the next phase is metaphase 1, and once again, I'm so sorry for the drawing. Oh, I was going to show you. I didn't draw the book had a picture of like what occurs for like description of each phase in anaphase 1. I didn't draw it, but I was gonna go ahead and show you what it is. So like, you know, you have your lipotine, and then your zygotine, pacotine, diplotine, and diakinesis. So metaphase 1 is when the bivalents attach via their kinetochores to spindle microtubules and migrate to the spindle equator. Presence of paired homologous chromosomes at the spindle equator during metaphase 1 is the crucial difference between meiosis 1 and mitotic division, because in meiosis it's the bivalents that are at the metaphase equator or the spindle equator, whereas in mitosis it's sister chromatids. So that's another difference that is between mitosis and meiosis. So the next one is anaphase 1. At the beginning, members of each pair of homologous chromosomes separate from each other and start migrating toward opposite spindle poles, pulled by kinetochore microtubules. The difference between anaphase 1 and mitotic anaphase is, once again, in anaphase 1, it's the bivalents that are being separated, whereas in mitotic anaphase, it's the sister chromatids that are being separated. To prevent chromatin separation from taking place, though, while the bivalents are being separated, there's a special protein called shugashin, that protects the cohesions located at the chromosomal centromere from being degraded by separase. This allows for it to the sister chromatids to stay intact. So the final stage is telophase 1, and as you can see, it's kind of the same drawing as mitosis. So telophase 1 is marked by arrival of haploid set of chromosomes at each spindle pole, which bivalent ends at what polar end depends on how the bivalent happened to be oriented at the spindle equator. So there's no rhyme or reason at which way they go. Once they are at each end, nuclear envelopes sometimes form around the chromosomes before cytokinesis ensues, generating 2 haploid cells whose chromosomes consist of sister chromatids. So that's the end of meiosis 1. Meiosis 2 is literally the same exact thing that occurs in mitosis minus the prometaphase step. The purpose of meiosis 2 is that of mitotic division, to parcel the sister chromatids created by this initial round of DNA replication into 2 newly forming cells. Meiosis 2 is known as the separation division. I really didn't make another drawing for meiosis 2 because it's the same thing as mitosis, and since I already went through it, I didn't think I should draw it again since it's at the beginning of this video. But yeah, that's basically it, so thank you.